a basic immunologist and a translational immunologist, you highlighted very nicely this idea that immune systems are really dynamic events and multicellular events. The T cell is an important cell in the initiation and final outcome of that immune response. What are the other cell types that you think, as we move forward in cellular therapy, that we might want to also involve in that? And is there a role for things like synthetic biology in initiating these events?
Yes, I think synthetic biology helps us to understand the different components, how they interact, and allows us to take parts of that biology and synthesize it into something new. But the complexity is considerable. So we're looking at two main types of different cell type to assist T cell function. First is invariant chain NK T cells, which have much better ability to traffic within tumors than conventional T cells and recognize different types of molecule in association not with MHC molecules but with C1d and on monocytes and other antigen-presenting cells. Those look very powerful in their ability to change the shape of the immune response that occurs within a tumor, so that if you start attacking a tumor, it produces a whole lot of defense mechanisms like myeloid dendritic cells and tumor-associated macrophages that turn off the rest of the immune system. By using iNKT cells, you can interrupt that dynamic change and make sure that the tumor doesn't evade the attack by the primary T cell. And the second area that we're looking at is NK cells, which do seem to be resistant to being rejected by the host and which do not themselves cause graft versus host disease. So these may be very useful initiators of an immune response that you can then follow up with the T cell. So the complexity is high, but I think, as you said, if you adopt a systems biology approach and understand what bits are influencing which other bits, you can come and get the key components, the key opinion leaders, if you like, and feed them in at the right time.
That's really well put. So from a fundamental scientific standpoint, that makes enormous amounts of sense. At what point do we begin to get regulatory buy-in to try these kind of trials that can bring in other cell types and initiate system-wide responses?
So I think you've hit on a major question. I think the two difficulties are how do you regulate, how do you develop studies that use multiple cell types, some of which will never have been used in humans. How do you develop the studies that will tell you whether those are safe and effective? How do you pay for those expensive studies? And how do you get the companies to work together, each of whom owns a different piece of the action, and none of whom are too keen to devalue their piece in order that somebody else can use theirs. And those are difficult questions to which I think we don't yet have an answer. I think we can begin to see how the answers are going to evolve with the checkpoint antibodies used in combination with cell therapies and used in combination with small molecule drugs and so on. But that's taken a little while to emerge. I think what will push it is success. I think as people find things that work, combinations that work, there'll be an impetus, a drive to try other things and see if they work. But without that, it's going to be difficult, no question.
If you looked at the entire space, whether it's virus-specific T cells or CAR T therapy, oncolytic viruses, checkpoint inhibition, what do you see as the next promising combination, even therapy, that will be seeing some kind of effect in the clinic, whether it's cytokine biology or what is it, trying to initiate that system-wide response that you mentioned?
I think there's a lot of areas that could work. The problem we have, as everybody knows, is that once you start having combinations of things, you essentially have no good way of modeling them. So you can find a model that predicts something will work, you try it in humans and it doesn't, you try something in humans that works, you go back to a model and you find it didn't work in that model. I'll just give you one example. When we started our EBV T cell trials to treat immunoblastic lymphoma in patients after transplant, there was no animal model. We went straight from the dish, so to speak, into the humans, and it worked beautifully, very well. About 12 years later, when someone finally developed an animal model, we tried it in the animal model, and all the animals died of severe graft versus host disease. If we'd had that animal model, we'd never have used it on the patients. So I think that remains a huge handicap to anyone who wants to try combinations. You can always come up with arguments for why they should be used and find studies in vitro and in vivo that suggest the positive outcomes, but the reality is that these have to be sequential, iterative studies. You come up with an idea, you try it out in a mouse model, if you have one, you try it in a patient, but it's really only in the patient that you can find the answer. I think things will get better because the depth of analysis that's now possible of events in vivo, in patients, gives so much more information that it gets much easier to predict back what's going to work and what changes you need.
Right.
But it's tough. That's a very hard question.
To your point, exactly. You have to study the system and really to understand the system itself.
Yes.
And the opportunity to do that, I think the clinical trials that you and your team have done really provide a huge opportunity for people to immune monitor, to really get an understanding of what the mechanism of action is when it does succeed,
Yes.
what the mechanism of resistance is. And then also to maybe look at other fields, like if you're studying allogeneic field, we could look at things like bone marrow transplantation and successful engraftment there to look at mechanism.
I think regulatory agencies are more and more aware of that problem. They're very willing to allow these kinds of adaptive designs.
Thank you, Malcolm. This has been a great conversation, and I think, certainly speaking for myself, I've learned an enormous amount, and hope that the audience has as well. Thank you for taking the
Well,
time.
thank you. I always enjoy speaking with you, John.
Appreciate it.
Very impressed.